right. Okay. So good morning, everyone. Thank you for being here this early this morning. We have a very esteemed speaker from University Kebangsaan Malaysia. His name is Associate Professor Dr. Firdaus Raih. We were classmates as undergraduates in cell and molecular biology at Faculty Science Hayat many years ago. Dr. Firdaus then proceeded to do his master's in UKM before then going on to do his PhD in the University of Sheffield. He has published extensively in the field of bioinformatics. And in particular, there was a publication in the journal Science not too long ago. And today we are very lucky to have him on board. Perhaps even be able to shed some light on the COVID-19. So the floor is yours, Associate Professor Dr. Firdaus Raih.
Thank you, Dr. Aisha. First of all, I'd like to thank the Faculty of Medicine, Professor Lau, Dr. Aisha, for inviting me to give the talk today. I'd also like to thank Mr. Shams from the Orthopedics Department for arranging the technical aspects of the talk this morning. Good morning, everyone. Let me just share my screen. So I hope everyone's able to see that. So as introduced by Dr. Aisha just now, my name is Firdaus. I'm from the Department of Physics at Faculty of Science and Technology, UKM, and also with the Institute of Systems Biology. And I would like to begin this morning by this statement that biology is awash with data. And the majority of that data in biology comes from DNA sequencing. And for those of you who have not or are not familiar with DNA sequencing, basically it's just a string of characters that represents their DNA bases. And in this case here, you can see an example here of the string of bases. And today there are currently about 200 over million sequences available in public databases. And from this sequence, we can actually translate that DNA into the protein sequence. And the abundance of these sequences means that there are many of them that we can use to compare. And it is perhaps the most common analysis in bioinformatics. Let me remove this for a while. And in bioinformatics, the most common way of getting or assigning function is to compare between different sequences. So what we do is basically use alignments to understand the functions of biological macromolecules. Now that is fine because there are rules that allow for such extrapolations. So if you have one sequence, it is similar with another sequence. Because of these rules related to protein folding, we can say that if a sequence is similar to another, therefore the function is also similar. Okay? But we must take into consideration that proteins do not look like this. It is not one-dimensional. Proteins instead, they take up a 3D form and shape. And that 3D form allows them to carry out their functions. So instead of looking very one-dimensional, a series of letters, the proteins actually look like this. The sequences do not provide the structural context and therefore does not allow for direct insights into the function that the protein may have. We also know that the structures are conserved over evolutionary time long after the sequences have diverged. What this means is that you can have very dissimilar sequence or different DNA and protein sequences, but the structure can remain very similar. So these structures are archived in a database called the Protein Data Bank. It is basically a repository of the coordinate data for biological macromolecules. So the coordinate data is basically just Cartesian coordinates, where you've got the atoms of the amino acids in 3D space being placed in X, Y, and Z. So it's basically similar to your X and Y graphs. Instead, here we have a third dimension, which is Z. So in this example here, you can basically see the atoms for valine and the positions of those atoms for valine in 3D space. So for many of us, we've probably seen pictures or renderings of molecular structure data, including the coronavirus protein. And that's basically extracting this information here, the X, Y, Z, and allowing a computer to render that 3D shape. So there are about 173,000 structures available in the PDB since its inception in 1971. Okay, and of that, 921 of them are SARS-CoV-2 structures. Now, this is quite amazing because in a very short span of time, 921 structures associated with one disease and one organism have been generated. This is very seldom the case. Usually, structural biology is a very slow process. Despite this very impressive rate of data acquisition, Senator Sanders, he doesn't look very impressed with it. So to find something meaningful or insightful or useful in all that data, it's like finding a needle in a haystack. Now, I would just like to focus the talk on structure data. So we know that for some of us, let's go back to undergraduate biochemistry. We know that similar 3D arrangements in amino acids can lead to similar chemical activity. In this case here, we've got a serine protease catalytic triad in two unrelated enzymes, chymotrypsin or trypsin and satylysin. They are totally unrelated in terms of evolution. The structure that they take up are also very different. But yet, you can see this similarity of how the serine protease catalytic site is arranged. And that allows for it to carry out a similar mechanism. And again, here I demonstrate that although we can superimpose the serine protease catalytic site, the structures cannot be superimposed very well. So if we look at it here, it's all a mess and it's not overlapping as nicely as the catalytic site is. Now, I want to go and demonstrate why understanding that substructures is very important in biology. So let's take here, we have a protein structure of an enzyme, and that colored bits there is the catalytic site. Now let's remove the surface so that we can see it in a simpler form. And this here is a rendering of the secondary structures. So it's quite often we see proteins looking like this or rendered as the secondary structures. Now let's look at there is the active site of this enzyme. And I've removed the background, the secondary structure, so that we can see it more clearly. Okay. Now I'm going to add back the backbone so that we can now see it in context to the rest of the protein structure. Now I add in all the other amino acid residues. So as you can see quickly here, the active site just now has suddenly disappeared or lost into the background. Now let's zoom it a bit and zoom it some more and zoom it some more. So it's clear now that because of the similarity of all these atoms, if we were to visually try to identify motifs or catalytic site, we would be lost in this haystack. And now I'm reducing it back to the backbone of the protein so that we can again have a look at that active site. So there it is again. And I've just removed the backbone now so that we can see this even more clearly. Now I've put back all the atoms, and again, as quickly as although we know it's still there, it's very, very hard to discern the active site from the rest of the background. And if many of you can see it, I've colored it slightly differently. Even that does not really help because it still goes right into the background. So that's for proteins. What about other biological macromolecules like RNA? Is it easier to view them, or is it going to be just as difficult, or perhaps even more difficult? So here, what I'm showing is the ribosomal, the large subunit of the ribosome, which is basically the macromolecule responsible for protein synthesis in all organisms. And it's clear here that the similarity of the shapes or structure of the bases to each other totally makes any visualization very, very difficult. So everything gets lost in this mess of atoms. So to find a specific pattern or motif in one structure is akin to finding a needle in a haystack. Now we know that there are in excess of 170,000 structures. So finding a specific pattern or motif in more than 170,000 structures is like finding a needle in many haystacks. And unfortunately, not all the haystacks have the needle that you're trying to find. So how can we find a needle in many haystacks quickly and cheaply? To do that, we turn to a branch of mathematics called graph theory. It's basically a branch of discrete mathematics. I typically, unless it's an information science or a computer science crowd, I don't really talk about the graph theory, so don't worry about it. No equations whatsoever. Um, but graph theory, I don't know how many of you have heard of it. It's actually something that almost everyone here will probably have used at least once. If you're from the Faculty of Medicine, probably at least one. You have probably done a Google search. You have probably used ways to get somewhere, and you have probably been suggested friends on Facebook. All these use graph theory. So in graph theory, a graph is said to consist of nodes, also called vertices, vertex, uh, and edges. And it is these edges that describe the relationship between the nodes. So imagine your Waze program. The nodes can be destination points, and the edges can be the roads that connect these destination points. So you might want to figure out which ones is the closest or the fastest that will get you from point A to point B. Uh, you can also have vectors to these nodes. That means they have direction. And you can also have additional data incorporated into the edges. For example, not only distance, but you also want to know whether a road has tolls. And clearly that's possible when you use something, an application like Waze. Now let's get back to our example of the serine protease catalytic triad. Now I would like to dim that a bit and try and add this and represent the side chains of these amino acids as dots. Now, what does that look like? They're basically the nodes that we looked at just now. And the distances between these nodes are actually the edges of a graph. So here we can see that an amino acid arrangement in three-dimensional space can be represented as a graph. Let's go back to that example just now. So this substructure of the catalytic site can also be represented as a graph. And how do we represent all these amino acids side chains as graphs? Basically, we take 2 points and represent an amino acid as what we call a pseudo-atom vector. Why? Because we take 2 points, give them a direction. So for example, in this case, to the right, an arrow to the right, a direction to the right, and the distance between them the space that this takes up becomes an atom, but not a real atom, a pseudo atom that represents, in this case, cytosine. Sorry, cysteine. Okay, and in this case, this represents an aspartic acid. And going back to that serine protease catalytic triad, here uh, a, 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 a better view where you can see that this side chain of the amino acid is being represented as a graph. So you've got a pseudoatom vector there, and this becomes a graph. And these distances are what define the edges of this graph. How we extract the distances is quite easy. We use Pythagoras geometry. So basically, it's the stuff that we learn in mathematics during our high school days. So we can use this substructure as an input to search for other protein structures. Let's say we're interested to find occurrences of this substructure in other protein structures like this. So we take this as an input and we search for it in the rest of the structure. So this can be a graph. The rest of the structure can also be made into a graph. And the matching of these substructures to occurrences in other structures, we can compute them using very fast algorithms that are available. And these algorithms solve a problem called the subgraph isomorphism problem. So basically what the algorithm does is it takes a subgraph and, it takes a graph and finds whether that graph exists as a subgraph in a larger graph or in a larger set of nodes and vertices. And that allows us to search for protein structures for the occurrences that we are interested in. The example that we used just now is that catalytic site of a serine and an aspartic acid. Now, for those of you who are familiar with this sort of structure searching, you would say that surely that is an obvious problem and a rather obvious solution. So if you've got something like that and you understand a bit of graph theory and mathematics, you say that, yeah, what's so great about that? That's nothing at all. What's great about it, if I can say that part of work, is that most other methods actually use the C alpha backbone. So most other methods use the backbone of the protein as the graph. We are probably one of the only methods that use the side chain of the amino acid as a graph representation. Now, why is that important? Because as this example clearly demonstrates, in some cases, the side chains superimpose quite well, but the backbone or the C alphas do not superimpose. Now, why is this important? It's important because we know that the side chains are the business end of the amino acid. Okay? It is the side chain that is going to do the catalytic mechanism. It is the side chains that are going to do the binding. So if we were to look at it in a functional perspective, this would be a very functional superposition. Whereas if we were to try and look at from a functional perspective also, the C alpha would not reveal this functional superimposition. So the methods that we have developed are useless if we're the only ones able to use it. So we have made them available as web services so that other people can use it also. So they're using those same methods. There are two programs available. One takes a whole protein structure and searches for motifs inside that protein structure. The other one, like the example just now, takes an input motif and searches for that input motif in a database. All right, one example I can give you is that you have a new protein structure and you're wondering, what are the known motifs inside that protein structure? So you can use this program called Sprite, and it searches a motif database to identify uh, what structures are in that particular, what motifs are in that particular structure. Once you've found an interesting motif, then you might want to try and see whether these motifs are available in other structures as well, especially structures that do not seem related. Now, we were curious whether those same methods would be applicable for RNA structures, and yes, we were able to prove that we could also use them for RNA structures. But I'm not going to dwell on the RNA structures. We just show that by doing that, we could use the information we collect for the design of synthetic nucleic acid constructs. And this is actually uh, in collaboration with a former University Malaya uh, colleague of yours, perhaps Dr. Efirul Ikhwan Ramlan. So the point here is that we can actually find the needles in the haystacks. But what can we do with that? One thing that we can do with that is we want to find similar substructures in unrelated proteins. And perhaps this can help identify or assign functions to proteins that have unknown functions. This is quite often the case where you have a gene, but you haven't figured out what that gene may be doing. Okay, we can identify these genes from transcription experiments like RNA sequencing. So we know that it's a gene, but we have no idea what that gene is doing. When we search a database using the sequence, it's not going to reveal, or it hasn't revealed anything insightful. There's nothing similar that allows you to say that, oh, this gene is similar to another known gene. Therefore, you could say that the function is similar. So you try and get the structure, and from the structure, you still can't figure out what it's doing. This is quite often the case with us. So part of my lab works on structural biology. So we do protein crystallography. And in this particular case, we found a similar arrangement of an aspartate serine that forms this catalytic site. And this catalytic site is similar to Cas2 nucleases. But the sequence similarity does not allow it to be associated with Cas2 sequences. So it's quite divergent in that sense. But the structural evidence allows us to propose that perhaps this is a new family of Cas2 proteins. And this is actually, we have submitted this for publication for nucleic acids research. So yes, we can annotate or assign functions to proteins that have unknown functions. But let's try something important, perhaps in a medical sense. Can we use it for drug repositioning? At times, this is also called drug repurposing, but there is a specific reason why I use the term drug repositioning here. So using that similar concept of searching for 3D amino acid arrangements or 3D motifs, we can take known drug binding sites and use that program to find similar sites in other structures. So what we want to do is find a site that looks like or is similar to a known drug binding site in a protein that is not known to be a drug target for that particular drug. And we were able to carry out that particular search. And again, we developed it into a web application called Drug Reposer, where it finds similar arrangement of amino acid side chains and identifies this as possible sites for drug repositioning. in a sense that you take a known drug like this one and you try to reposition it into a new site in a protein that was not known to be a drug target. Okay, so in this particular way, approved drug compounds can be rapidly repurposed to treat other indications or diseases. So we're wondering, can drug reposer be used for identifying target sites associated with specific diseases? So what we did originally was just a proof of concept. We showed examples that we can find drug binding sites in proteins that are totally unrelated to the original drug target. But we didn't really do any deep work into it. But now we wanted to explore it further. Can we use it for COVID-19, for example? Can we use it for cancer? Can we use it for antimicrobial resistance? So someone at IMR became interested in it. So he is trying to use it to find to repurpose drugs for cancer. We are actually trying to see whether we can use it for antimicrobial resistance. But this is the problem that we are faced now, COVID-19. And doing the analysis, we found that we were actually able to find targets that are known drug binding sites in the structures of SARS-CoV-2 proteins. Okay, and just now I showed that there are 921 SARS-CoV-2 structures. So those were the ones that we use to identify or as targets to find whether there are known drug binding sites in them. And this is some of the work that we had published in October or was it September of last year, where we were able to find proteins of SARS-CoV-2 that could potentially bind to known drug targets. Okay. And one particular target protein is the SARS-CoV-2 ADP ribose phosphatase. This is interesting because this one particular enzyme has multiple or binding sites, possible binding sites to multiple drugs. So this is chloroquine. It can bind to chloroquine. This is colchicine. This became sort of, how shall I say, viral past few days from that Canadian study. Darunavir, ritonavir, and a host of other drugs from HIV work. So here, it sort of provides a mechanistic explanation why a drug like colchicine might be able to work. We don't have any validation at the moment that this is actually the case. And what we are expressed about, or what we are clear about in this work, is that this is not a clinical interface. That means you're not supposed to use this for finding drugs directly to treat patients, but what this is for is to identify drugs that we can use for trials, repurposing trials. So because we have the capacity to find similar binding sites that are similar to that of known drug binding sites, however, the problem with that is that by binding to an unintended target, especially in the host, that may cause other effects, including toxicity. So one thing that we decided to do was see whether we could use the drug reposer search engine as also a toxicity or off-target binding indicator. And yes, we were able to do that, where we could find a site in proteins, for example, that are that, oh, sorry, there's some things that pop up here. Let me just remove it a bit. I can't see the screen. We could use the same method to identify potential off-target sites, especially in host proteins. So that's not what we want to happen. Okay, and examples of off-target sites. So we've identified the compounds that could potentially be used for SARS-CoV-2 proteins, and then we tried to find whether there are similar drug binding sites in human proteins, and we were able to find those. But this is not necessarily a bad thing, because after all, these drugs had already been approved for use. And the fact that we can find alternative or off-target sites in human proteins does not discount the drug as a potential repurposed therapeutic. What is important here is perhaps that it can provide the necessary intelligence to clinicians and trial programs to avoid what I call surprises and allow them to mitigate side effects or assess the cost benefits of the off-target effects. So early on from the program that you want to do the repurposing, you might already be able to predict possible side effects that you might see in the trial subjects. So we hope that as the more structural data becomes available, this capability will improve and become more accurate. Okay. Another thing that we explored was the fact that if we have a known drug compound, perhaps a compound that is similar to that in terms of structure could also be used. This has been shown in some cases where slightly changing the compounds helps a lot, because some people may not be, the efficacy of one compound may not be good for others, but for some others, you know, compound A is good for this person, but compound A doesn't work for another person. So, but compound B, which is very similar to compound A, might work for that other person. So you have a sort of wide range of compounds or weapons that you can use against a particular target. Okay. So that's what we have found. We were able to also find alternative compounds that perhaps can be used or can be subjected to drug trials. And these are examples of those compounds. For example, here, the compounds have very minute differences. So you've got almost similar compound there, except that difference there, almost similar compound there, except that difference there. Okay, and sometimes these minute differences could be the difference between efficacy and non-efficacy, or allergy and non-allergy. Okay, so we could use it for drug repositioning. So the thing that we wanted to do next was we didn't know what else to do next, so we just thought, let's just explore and see what we could find using this program. So computational biologists perhaps have too much time in front of their on their hands in front of a computer, because that's our job. So we explored the problem with SM, for example, is to find a known motif, okay, in existing structures. So we need to know something. We need to know what we are looking for. What if we don't know what we're looking for, but we have an idea of what it should be? For example, we might want something that has a serine and an acidic residue. So we know that a serine and an acidic residue could potentially function as an active site, but we don't know what it looks like. So that way we can try and discover something new. And this is what we managed to do, we developed a sort of a wildcard search engine, and we called it Imagine. So it stands for Imagine an Amino Acid 3D Arrangement Search Engine. And we were able to do quite a lot of interesting things with Imagine by simply imagining what we wanted to find. So we wanted to find, oh, can we find, for example, a cluster of histidines very close by to each other? Or can we find DH, histidine, aspartic acid, histidine, aspartic acid, histidine, aspartic acid within a certain distance of each other? Okay, so it's basically trying to uh, explore and see whether we could find something new that we didn't really know about in the first place. Okay, and interestingly, we were able to find examples of those. So it was quite by surprise at first, because some of the things that we found had not been reported before. So it became a treasure trove of information that we found most data, most stuff that we could, or we had the hands to process. So these programs had the ability to identify conserved 3D motifs uh, that result in the conservation of function in totally unrelated proteins. Example here, you can see this metal binding site, but as you can see, the proteins are not related at all. They're totally different proteins. One is in tan and one is in blue. But you can see that the metal binding site is actually the same. It's exactly the same. Okay, so that's interesting. Uh, we are in a way able to find novel motifs that are not going to be detectable in a sequence search. So these motifs are detectable only in a three-dimensional context. And many of these novel motifs are hiding in plain sight. So basically you're looking at them, but you don't know that it's a motif because you do not have the right context to allow you to say that it is a motif. And this is one example that I like to show. This is actually consists of a multimeric protein. So many subunits assemble to become this protein. And this is one subunit. So at sequence level, what we can see here is that you've got an arginine here, aligned, and a lysine aligned. So arginine and lysine are both positive amino acid side chains, positively charged amino acid side chains. But in the sequence, it is one residue. So you cannot call one residue a motif, but within the context of the structure, it is clearly a motif or something that is preserved. What we want to do next is we want to try to use machine learning to transfer this knowledge to sequence level searches, because it is the sequence level searches that most people use. Okay. And again, when we explored that, we found that a lot of these motifs are at the interface of uh, molecules, asymmetric units. That means proteins that are interacting in a quaternary structure. This example here, you've got this histidine, amino acid histidine. There's 6 of them, okay, that helps coordinate this metal site. So it binds to this metal. But in terms of the genetics, that protein is encoded by only one structural gene. Okay, and it assembles into this quaternary structure, and only then does this motif take shape. So here, all you can see are 2 histidines. But once you have the context of the full assembly, you can see that, oh, this is actually a biological interface. So it acts as the interface of quaternary assembly. And again, since we saw that we could do this, we made a database out of it called the Intermolecular Context Biological Assemblies Motifs Database, ICBMs. And being having grown up in the Cold War, ICBMs to me meant an intercontinental ballistic missiles. Okay. Right. In conclusion, 3D substructure searches. Sorry. 3D. I'm trying to remove this a bit so that I can see what's going on. 3D substructure searches in biological macromolecules such as proteins and nucleic acids can be carried out by representing the amino acid side chains or the bases as graphs. Okay, and these searches can allow for the needles or these 3D motifs to be found in the haystacks that are the protein structures. And being able to find these gems, hidden gems, has allowed us to carry out the annotation of functions in uncharacterized proteins. It has also allowed us to identify alternative targets for the repositioning of approved drugs. And it also has also led to the discovery of novel interface motifs that we hope one day can contribute to the deeper understanding of how proteins can assemble into quaternary structures. And before I end the talk, I'd just like to acknowledge my collaborators over the years in bioinformatics and in structural biology, and the funding that I've received over the years as well. I also like to thank computing resources at the Malaysia Genome Institute and USM for hosting our web servers. And of course, none of this would have been possible without the forced labor or the labor camp inmates who are hard at work trying to make all this possible. I'm quite happy and pleased to say that many of these inmates have been released for good behavior. With that, thank you for your attention.
Thank you very much for that very insightful talk, Associate Professor Dr. Firdaus. We have a number of questions this morning, and many of them are actually from, well, we'll begin with Professor Tengku Sarah from the Orthopedics Department. So if I'm looking at this correctly, among the questions is that could there be a site for the drug ivermectin in COVID-2? So what's your thoughts on this?
We could check. I can't remember all the sites that we found, but the work has been published. So you could, well, you can email me if you want to know, or you could look at the paper, or you could easily check the database itself. So we make the database available. It's MFR Lab. I think I showed it just now. .org/drug-reposer/COVID-19.
Yeah, so it's actually very easy to find Dr. Firdaus online. You just Google his name and you'll get his email. His email's all over the internet.
Okay, that's not good.
All right, so I think it's a safe conclusion that your databases is a cut short method, I think, to find useful drugs. Yes.
It is useful for identifying sites for known drugs, but we think it can also be useful for developing new drugs that can target known sites. So perhaps, if an existing drug is not as effective, you could then rationally design a chemical compound that could perhaps better bind to that target. Right. So it's useful for rational drug design as well.
Would we have that capacity in Malaysia? What's your thought on this?
We can easily do that because you can easily collaborate with a synthetic chemist who can design that molecule and they can easily derive an existing compound to become another compound. So we've done it before in that way.
Right. So that's really good to know. Okay, you have a rather long question from an anonymous attendee. So I'm just going to read it out. Thank you, Doctor, for the insightful talk. I have some questions. To classify a protease, we usually look upon the conserved catalytic residues, for example, asparagine, serine, histidine, but in a protein, sometimes there are more than one conserved asparagine, serine, and histidine residues in the polypeptide chain. How do we know which one are considered as catalytic residues, and is there any software, web server available to help detect those?
Okay, I think there could be a few aspects to that question. I mean, a few answers to that. One, classifying the proteins in terms of the sequences, you can easily use an existing database. I think one very classical database that has been used for more than 20 years for proteases is called MEROPS. Uh,
Sorry, how do you spell that?
MEROPS,
Right.
PS.
Okay.
I can't remember what it stands for, but
P for Thailand. Is it?
M M M E R
MEROT.
O P S P.
Oh, MEROPS.
Uh,
Oh, okay, okay, okay.
okay, and you can also use a structured search. It depends on what information you are using as a starting point. So if you have a sequence, then it could be difficult to identify the catalytic site, as you have rightly mentioned, because sometimes you may have a protease that is unknown. It could be new, okay, and you cannot find any sequence similarity. So the key to that is identifying the catalytic site. So one way that we as structural biologists do it is by determining the structure, and from the structure we look at whether it looks like a catalytic site, and at the end of the day, to confirm whether it is or not, we have to do the assays and mutate that site. Um, so it's not a matter of just inputting in something. If you want to validate it, you need to do a bit of lab work as well. Um, Not sure.
And you have that capacity in your lab. You mentioned the crystallography that you do in your lab.
I do have some of my students who work on the structural biology aspects of it. Uh, but for proteases, I work with a collaborator of mine, David Rice at Sheffield. And we have actually just, for an asparaginase, we believe that for the past 50 years, they've got the geometry of the site slightly wrong. So we might have evidence that could rewrite the geometry of the site, and we're still trying to figure out whether our results are actually correct. So we're doing experiments and some computations for it. So
Right. So the work that you're doing is really, I saw that website that you had on the drug reposer. So is that something new that's been built by your lab?
yeah, Drug Reposer is new. It only came out in 2019, so it was published in 2019. It sort of, when we were looking at these things, we found that a lot of the things ASAM retrieve are binding sites. So one thing led to another. If it's a binding site, could it be a drug binding site? Since it's a drug binding site, can we find other drug binding sites? And if we could find other drug binding sites, can we make it a database? And hence, since we could make it a database, can we make it searchable? If it is searchable, then we might as well let others use it as well. So that's how Drug Reposer came to be.
So are you saying that there isn't anything quite like this yet, drug reposer
Uh,
available
there
online?
are many drug repurposing processes software, but not the way we do it, in the sense that we look at the side chain similarities. So others, including the ones published for SARS-CoV-2, looks at it in terms of the pathways. So if it's a related pathway, can we target that related pathway in another organism? So you try to repurpose similar pathways. But what we're trying to look at is binding sites using the side chain similarities. And this is quite unique, and we almost have a monopoly of how to do this at the moment. So, matter of fact, last night, someone from the Weizmann Institute was saying that our Israeli, yeah. Someone from the Weizmann Institute was saying that our website seems to be down a lot because it's not a very powerful server. It's a very low end server, and he was saying that, would you like us to host it for you? Because they're sort of our hardcore users.
Right. Are you gonna take him up on that offer?
We'll see.
Okay, that's really good to know. So I don't see any more. Oh, there is one more. Um, right. So there's a question from my head of department. Can the software be used for protein-protein interaction studies? For example, antibody antigen binding.
Yes, it can. As long as we know what the starting point is. For example, if we know where the a known antigen binding site is, or what we believe the antigen binding site to be, we need a starting point, or we need an understanding of what that starting point might look like. So yes, it can be used for that. For protein-protein interaction studies, we do actually use it for protein-protein interaction studies, but not for predicting a protein interaction interface, but to classify protein interaction interfaces. Like I showed just now, since we could find the intermolecular contacts that are very conserved, uh, we can then perhaps try and classify all the known intermolecular contacts. And hopefully by being able to do that, we can in the future understand why some proteins will interact and interface and why some proteins will not do so. So that could also lead to engineering of these large assemblies, since we know we could engineer interacting sites. So you have a protein doing one thing, a protease, and another protein doing DNA binding. Uh, Naturally, they wouldn't interact, but if we can engineer an interacting site because we know what it should be, we can then make them interact. So
But you don't
that's
actually have this functional thing going on in your laboratories, right?
No. So a lot of the functional stuff that we do tries to understand novel functions. So we try to find new structures of that have not yet been characterized. So we purposely go through databases to find genes that are annotated as hypothetical proteins. So we take that and we try and figure out what the function of these hypothetical proteins are. Um, we also do a lot of nucleic acid assembly work. So what
Mm.
we try to do is engineer synthetic nucleic acid structures, and these structures can self-assemble. So hopefully we can one day design. It's been done actually. So we are quite behind in that aspect. We can design cages uh, that could perhaps hold drugs. So you can deliver drugs to specific binding specific sites by engineering DNA nanocages, RNA delivery vehicles, and things like that. We've not really done anything in terms of practice except for proving the concept that it works. So we've done it. We can show that we can whimsically design an RNA structure, a DNA structure, and we prove that that whimsical design actually ends up into the structure that we design. And we have proven that using atomic force microscopy.
Okay. So sorry.
There's a question here that asks me about my name.
Yeah, I
Is it a
purposely didn't
hyper
mention that.
uh, advantages? Oh, no, no advantages. It's just that well, Raih is my father's name, so it's sort of strange when by doing that, it makes it my name. It becomes my name. That's all.
Yeah, you just don't want to lose the Muhammad. I mean, it's the
Yeah,
same as
yeah.
mine. I suppose this is what happens when you get trained in England. Like I have a dash hyphenated
Yeah.
Abdul dash Aziz. Anyways, moving on. Last question would be coming from me, I reckon. You very interestingly mentioned the 921 sites.
Yeah.
Yeah,
Structures.
yeah,
921
yeah,
structures
yeah.
of
Sorry, yeah,
SARS-CoV-2.
yeah. And then you took a dig at Bernie Sanders.
Yeah.
So evolutionarily, why do you think this happens?
Okay, I think that I must explain that a bit. There are 921 structures there,
Yeah,
but there are actually only about 30 proteins for SARS-CoV-2. So the reason why structural biologists do that is they like to create uh, multiple coordinates of the same proteins. Why they, you stick one compound to it, one structure. You stick another compound to it, one structure. You bind it to something else, another structure. So although there are about 30 SARS-CoV-2 proteins encoded in the genome, I think there are about 26, 27 very well-known ones. There are 921. So that number doesn't make sense. It's actually some of those like proteases, I think there are tens of protease structures. It's of the same structures, but it might be bound or interacting with other things. So for example, you've got the RNA-dependent RNA polymerase. And you have got that on its own, you've got that bound to another protein, you've got that bound to RNA, you've got that bound to a drug. So at the end of the day, the same protein structure, you have a lot of examples of it. And hence why for a genome that encodes only for about 30 proteins, will end up having 921 structures. So all of those have they're distinct in a way that they might have different conformations, which will help us understand. They might have mutations. For example, some guy might have mutated D614G to understand what sort of changes it does to the spike, or any other mutation. So it could be the same proteins mutated many times. Hence, you might get hundreds of examples of the same proteins, but they're all mutants with only one that was the starting point. So yeah, you end up a lot.
Okay. So we can carry on, and it's good to know that we can carry on and have continuous geeky conversation on this. But then time's up. I would like to thank Associate Professor Muhammad Firdaus, especially on behalf of Department of Parasitology. And we're very glad to have you on board. And thank you, Breakfast FOM, for giving this slot to Department of Parasitology, Faculty of Medicine, UM. And so we thank you very much, and we hope that this can lead to greater collaboration between your good lab and other labs. Okay, thank you very much.
You're welcome. Thank you for having me.